Welcome to Foresight's Molecular Nanotechnology Group. Today we have Amijai Saragovi with us. He is at Baker Lab, and his work is at the intersection of protein design and semiconductor nanotechnology. And it's been a long time coming, this presentation, so we're really excited to have you on.
Thank you very much, Lydia, and hello everyone else. So just as a kind of a small introduction, I'm kind of at the end of my postdoc now in the Baker Lab. I'm transitioning to Lund University in Sweden, where I'm now in Sweden, basically. This is why there is light outside. So this is why the logo also from Sweden and not only from the Baker Lab. So today I'm going to tell you a bit the story of my research at Baker Lab, but also maybe what is my vision for the future. And it's maybe a good place to note that some of the things I'm doing now for the future is part of a vision that is encapsulated in this website in bio.design, financed by Vinnova. And this is the idea of starting to use e-waste as a source for making semiconductors. Okay, so I wanted to talk a bit about e-waste before I jump into the story. So e-waste is an emerging problem, but also an opportunity. So on one hand, we're generating a huge amount of e-waste every year. So about 7.8 kilogram of e-waste, according to the e-waste monitor UN reports done every year. Most of it is not being recycled in an okay way. So only, say, a quarter of it, less than a quarter of it is recycled in an okay way, which basically means that 62 billion kilograms is being dumped into our societies, our environment every year. So we're building the same problem we did with plastic, just with things that are super toxic, and we give up on a lot of precious metal. And the way to present it is that your mobile phone probably contains most of the periodic table. So all these resources that are now a bit in the news because everyone tried to make GPUs and chips are out there and we're not using them. Right, that's just another view. So you can see that almost half of the waste that is described in mass is metals that we can use. And just another figure to show that it's not getting better. Okay, so what can we do about it? And here where we start talking about proteins. Like a lot of other things, we can learn from nature. So some of it you perhaps know, like nature knows to organize inorganic material. For example, in the bones of all of us, there are inorganic material, crystalline inorganic materials that is organized by protein. So the first example in the top, you see an array of protein in glass or silica sponges. These are type of sponges, family of sponges that are basically covered in glass. They're beautiful if you want to Google them. And basically you have an array, super organized array of proteins that catalyze the formation of silica on it. So this is kind of an enzymatic reaction that lead to the formation of super organized silicas that kind of look, if you look at it, it's a resolution that you need to build a semiconductor chip in terms of the things are the way they're organized. And then in number two, bottom left, we can see nanomagnets in magnetotactic bacteria that are organized into a campus. So each one of those is about 48 nanometers, which is what you need to move from supermagnetism to magnetism. And these are also organized by a number of proteins that both control the redox and organize the crystals into the right size. So if you get rid of the proteins, you get some magnetite, but it's not organized into something useful. And in number three here, it's an example of beta-guanine. It's an organic crystal that is used in many organisms, but just to keep a long story short, this is the reason why if you have goldfish, they're gold, because they have guanine crystals organized to reflect only this color. And you have even eyes made of guanine for ocean. So yeah, the atoms, another example of more like 2D and 3D organization of architecture in the nanoscale. Okay, so now the fact that nature is doing things, yes, nature is amazing, but can we copy what nature is doing, and how can we do that? I'm going to talk a bit about protein design to those of you who are not familiar with it, and that kind of will give you the introduction to what I did in my work. If you want to learn more about protein design in general, and I think there was a Foresight lecture about this, but also a very good lecture is a Nobel Prize lecture, the last one from December, where David presented the entire field. Yeah, so proteins are basically doing everything in living systems around us. Structures, they're nanomachines producing, like replicating DNA, and a lot of other things. So these are native proteins. Okay. So in the last few decades, David and others tried to resolve two problems that are at the core of protein design. And this is what we try to do in the lab. So one of them is a structure prediction problem, where you have a sequence of a protein and you try to predict what will be the 3D structure of this sequence. So you need an oracle. A Nobel Prize for this was given to the DeepMind team for developing AlphaFold. Let's note here that we also published RosettaFold in parallel to them, but they won the prize for structure prediction. And this is an amazing tool, because then you can check whether the protein you designed actually fall to whatever you want it to do. The other problem is how to design. So here it's a reverse problem. You have a structure and you want to predict a sequence that will fold into the structure. For this work, David won a Nobel Prize for many different things that were done over the years. So this is the core of everything we do in the lab: prediction and design. And this is kind of a typical process of, you will later see like actual examples from my work, but this is how we work. We first use diffusion, which kind of work almost the same as the diffusion models for generating pictures. So diffusion know to take random atoms, randomly organized atoms, so basically noise, and generate a protein from them, something that is actually a protein. And then if you give this model restriction, you get the kind of proteins that you want. Then once you have a design, you can then give it to another model called Protein-MPNN and generate a sequence that will fold into this model. And you confirm this with AlphaFold or RosettaFold, that actually the sequence you generated is going to form the structure you need. Okay, so this is how it works. This is what we do day and night. We work on a computer. We try to get to the designs, and we get a DNA. Today you can print DNA, mix it with bacteria, grow the bacteria. Next day we drink coffee and we produce our proteins. Yeah, and I think most of you know this, but just the kind of the engineering problem we deal with can fit 50,000 times into a human hair. So interesting thing to work with, especially when you need to deal with both soft materials, protein, and semiconductors. What is the vision of my work and the effort I'm looking for in the future based on the research I did? So we have a lot of e-waste. Maybe we can design proteins in bacteria that will use the ions from this e-waste to form nanostructures that are actually functional, that you can organize and make things out of. So create a circular process that is extremely scalable, because bacteria can reproduce in a crazy rate, and also good for the environment. Okay, so let's talk a bit about my work now. Okay, so we start with the basic. How do we approach the problem? So there are minerals in nature, and there are proteins that are involved in organizing these minerals, but there are no proteins that organize zinc oxide or hematite, or, for the sake of it, from a more general view, other semiconductors. So we started with kind of basic principles and a lot of guesswork. So at the top, with number one, you see a situation that is actually happening a lot in solution. You have ions in a supersaturated state, which means that the concentration of the ions should result in the formation of a solid phase. However, because in order to become mass solid state, you need to first become a tiny nanoparticle, you end up with a funny situation where the surface of the nanoparticle is so big in respect to the bulk of the nanoparticle that the nanoparticle is unstable. So you're stuck in a loop where ions are forming tiny nuclei, and then they dissolve into the solution. And this can go for years before a solid phase will emerge. Depends obviously on the concentration and how saturated the solution is. If you're able to specifically target a facet of this tiny nanoparticle and bind it, Then you stabilize it by lowering the energy needed to form, and then it will continue and form a particle that is also related to the facet you're binding. So based on this basic principles, we came out with 3 assumptions on how to target our semiconductor material. The first one was maybe even naive. Okay, let's just, if the surface is positive, we would do a negative protein. And if we do it in the right periodicity, they will just match. The second one is slightly more complicated. The idea here is that you use different coordinating amino acid side chains to partially or fully not fully, but partially coordinate ions in the period of the target crystal. And this will allow you to stick to it. And finally, we thought that since we know that crystals also organize the water almost the same way as ice is organized, perhaps all the protein need to do is to organize the water molecules to bind it. So these were our 3 assumptions. So how do you deal with this when you have assumptions, and how do you find out a design that actually works? You design a lot of things and a screen to search which one is working. So to do this, we first created a lot of scaffolds. Scaffolds are just different kind of base shapes of a protein. They were all flat, or most of them were flat. They're repeating. And the idea is that some positions within the sequence stay fixed. They're not changing. So these positions will force the protein into the base structure. But we still have the positions that are flexible, that are variable, in this figure marked in yellow, that are used to basically change the surface chemistry. So once we had a lot of scaffolds, we started diversifying the chemistry based on our assumptions, coordinating residue, water ordering residue, charge residue, and so on. And you can also organize it. So the same composition of amino acids, you can organize in a periodic way that matches a lattice, or just mix them. And we didn't know, we had an assumption that periodicity will help, but maybe also a bit of chaotic organization will allow us to find something very interesting. So overall, I think we had 7,000 base design or something like this, but then we varied other things. So we designed like 35,000 proteins, and we expressed them on the surface of yeast. And the idea then was to use flow cytometry to run them in a flow cytometer, which kind of shoot laser on each yeast individually, and you can measure the scattering. And the idea was, okay, let's just mix the yeast with, let's say, zinc oxide nanoparticle and calibrate this a bit, and then run them through the flow cytometer. And because zinc oxide has very different refraction index than water, we should see a difference. So we did this, and we repeated it 3 times, every time enriching for things that had a higher scattering. And then we end up with these examples. So we had 2 designs that seemed to bind very well hematite. They all had coordinating residues, histidine and tyrosine and so on. We also found things that seem to bind very strongly to zinc oxide. So you see on the right-hand side, the left one of the right, beta-solenoids with a lot of histidines and glutamates, and on the right side of the right side, you see another protein, but this one then only have threonine. So we're quite curious if the mechanism there is to do with water ordering, because we know that threonines organize water through a clathorate mechanism. Okay, so we find things that seem to bind to the nanoparticles that we want, but the question is now whether these will actually template and allow to shorten the reaction time and form the semiconductor in a solution. So I'm going to explain about the methods we use here. So on the left, you see the assay we developed, and the right is just a common fingerprinting of the material. So let's start with the top, the hematite design. So what we did here is a little trick. So basically, hematite will be red, but then a lot of other things will also look red. We had a problem to very quickly identify, let's say in a 96 format, which conditions or proteins were doing anything. So what we did, we changed the reaction so the default output will be magnetite. And then we had a plate where underneath each, we had a magnet. So very quickly, we saw whether we form magnetite or something else. And you can measure it with a few different methods, but one thing you can measure is the level of red versus other things. And you can see that when we had nothing like a buffer or we had a lysozyme, very quickly you lose the redness. And I can tell you that also you see like ground black things around the magnet. But when we added our two unidentified protein, H7 and H9, we saw some effect with the H7 doing kind of a crazy effect. I was amazed to see then that if we look with Raman on the output of these experiments, so here in the button you have the fingerprint of standardized hematite and the fingerprint of magnetite. And I was amazed to see that the lysosome and the NEAT looked spot on the magnetite, and the H7 and the H9 that were selected with the hematite nanoparticles gave like something that really looks like hematite. So I was quite happy and excited about this. And then with zinc oxide, we have an easy life because it has photoilluminescence. It can produce a fluorescent signal. So the nice thing is that you create a supersaturating conditions. You add the protein, if they have an effect, then you can read it, the fluorescence or the photoilluminescence wavelengths. So here in black, you see the nanoparticles, which are the ideal template, and then the proteins are making an effect. In the button here, you see lysozyme and this buffer just doing nothing. We confirmed this with XRD this time to see that we have the right fingerprint for this material. Another funny thing is that we found out that if we take Heme7 and we put it on iron, we saw inhibition of the formation of rust. So you see that if we mix it with lysozyme, after 2 hours, these are buffers, so you have a lot of salt. The reaction happened quite quickly. You get a lot of rust, lysozyme or just buffer. But if we add our protein, we somehow inhibited this. We're not sure about the mechanism here, but perhaps it's to do with the protein binding it and forming small pieces of hematite or something like this. Okay, we found our first candidates. Can we start redesigning them to do perhaps something a bit more useful? So one thing we found out that actually what we call Z0, so one of the zinc oxide templates, was forming these fibers that look a bit like amyloids. We saw that this is messing up our reaction. At the bottom is that on the left that we took the motif from Z0 and we redesigned it to be more stable and better behaved. And if you can see ZAP3 here, in here, this is the time taken to see the photoilluminescence signal. So this was the initial design, and this is how much it improved. And actually, after the improvement, it was faster than the nanoparticles, which kind of was shocking. The nice thing about protein design is that once you have the motif, you can start putting it into shapes. So here are different shapes that we were able to use to put the motif. So you see a C3 here, where the motif is pointing inside into the confinement area. You see the same with C6. And you can also do an octahedral cage and a tube. We did quite a few of those. So let's focus on the cage. You will have to take my word that all of them are creating fluorescent signal or photoluminescent signal, even as oligomers. But here we characterize it with cryomicroscopy. You can see a micrograph here with black things growing on our cages. We found different kind of outcomes. We had empty cages, probably because we didn't super idealize our reaction. We see some that are full. We see also ones that are coming out of the cage. This is because the interface probably is sitting on the C3 and it's not restricted in the third dimension. And then we also saw a case where like two cages came together like through a merged zinc oxide motif. When we reconstruct everything, we found that we are identifying mass in exactly where we planned for it to grow in the middle of the C3. And you can see this mass here. And if you further refine it, you see the kind of the ordering of that. Okay, so what are the future steps? So first we can work in 1D. So we are working on confining the materials better inside proteins that will target the different facets of the expected crystal in a very defined way. We can also start moving to things that look more like a fiber or a nanowire that is used in a lot of applications. Next, we are already working on things that will be organized in 2D. So you can think about something cool where you have 2D organization of a shape, and then you can also control. So one of them can be zinc oxide, one of them can be hematite, one can be silica. So you can start controlling kind of the organization of the material in a very tiny nano level. And finally, a bit more into the future, you can organize 3D, something that kind of already look perhaps like what you would want from an advanced transistor or chip. You can also work with different materials. You can work with single material, but check a lot of different semiconductors, magnetic materials, and so on. So you can diversify the material with the same shape. You can also move forward and start creating junctions between shapes. So two different materials coming together. So let's say one template is making zinc oxide and another one hematite and so on to create like interesting electronics or optical structure. Finally, you can do very complex material and also a doping, control the doping and so on. So there is a lot of work to do in the future. And like the dream is to be able to do things that are very pristine, that they could serve as electronics. I think this is where we aim at, and could be produced hopefully in bacteria. So we can potentially move all the process, including mineralization, into bacteria and then have a clone that is generating it for us more into the future. And then another just direction of this is that once to integrate these systems into living system, you can start doing things like cable bacteria, where you have an antenna within a cell or a hybrid material that is functional inside a living cell, which is another direction aside from just doing electronics. Okay, I don't think it's relevant for many of the people that talk. Maybe there are some Europeans, so we're making hackathon this week in Sweden. If any of you is from Sweden, you can log into in bio.design and register. But I guess there are not a lot of people from Europe or from Sweden on the talk. And I think it's time for questions.
Awesome. Thank you so much. Is there anyone that has any questions in the audience? Ted.
So this is very exciting. And you surprised me at the end there with the idea of collecting the minerals and then having them do something in the form inside the protein cages, or a 3D array of protein cages. That is really cool. So one question is, how concentrated can the minerals get in water in order to be collected? What do you do? Just dissolve the e-waste with acid, or how do you prepare it, and how concentrated can it be?
Yeah, so actually we went to a few e-waste recycling companies and had a discussion with them to understand how can you use these materials. Their opinion was that you can definitely, with some acidity, leach most of the ions, and there are a lot of bacteria that live in this condition. Extremophile can the acidity of this. Yeah.
Neat. Very good.
Awesome. Prokop, you're up.
Yeah, hello. So yeah, it's very interesting and also a bit related to something which I would like to do, but I'm interested. So basically the idea is that you build a protein and which has some like primary structure, secondary structure, and then you use the tertiary structure to actually form some, let's say, assemblies of these crystals. And like from point of view of, let's say, efficiently using this structural information, it would be probably best if we can like get as much of the underlying information to the final structure. And so this would call for, because your proteins are more or less, as I understand, kind of balls, but if there would be some scaffold, like if this would be like linear structural proteins, this would really be possible to encode into this length segments like many different, basically, for example, as you say, heterogeneous material and so on. So is there some, because I don't know much about protein design, is there any fundamental obstacle of this, or is this something which you would like to do in some later future?
Yeah, okay. I think I understood your questions. First of all, things that we're working on right now. So we're now working on other things, including fibers and so on. I would just say that the protein serves in two different functions. So one, they serve as a template. Here, the template is basically reducing the energy needed to form the crystal. So this is a motif that we found during the screen. The second job of the protein is to confine the formation of the crystal. So for example, if you just have a fiber with the protein motif, the same, we actually ended up with things that look like fibers with Z0, the initial design. What happens is just you create like what I call GUC, non-controlled formation of material, which is not what you're trying to avoid. And you don't need a protein to create uncontrolled formation of mineral. So I think the trick is to very carefully place to get a really good motif and then lower the supersaturation and create things that are super defined.
Okay.
Hope I answered your question.
Maybe, but I will ask later. But
I don't know if the email was there, but you can write me an email or send me a message on LinkedIn or whatever. I'm happy to answer more questions. Yeah.
I saw a brief hand raise from Christian and then also a hand raise from iPhone.
Yeah, this is Adam Moyer at iPhone. Amijai, I had a question.
Yeah.
Does this only work in so I understand that it's super saturated before the particles form. Is this like a exothermic reaction that like drives downhill so then the particles stay if you took away the protein?
Yeah, first of all, maybe it wasn't clear, but we're working in a regime where we're not changing the thermodynamic of the solution, obviously. So we're not making magic here. So we're working in a regime where the material want to form, but it just stuck in a kinetic barrier. A bit like the same way an enzyme works. The reduction in energy is not through catalysis, is through the fact that we change the surface to volume ratio. So if you think about a surface is not a happy thing for a nanoparticle. It's organizing the water, it's creating lots of energetic cost. And if you bind the surface, you reduce this organization of water and you lower the cost of the surface. So if you have enough surface that you bond, this is how you lower the energy. Was I clear, Adam?
Yeah, I think that makes sense. I guess I was asking then, you can make particles that would be like, I guess, stable then, like when you take them out of the supersaturated solution, right? If you're making like some titanium dioxide, right?
Yes.
From straight up titanium, like it would oxidize and then, like, I guess, have you done any reactions like that where you can actually make particles then that you can keep around?
Yeah, so this will work. So you can keep them after the nanoparticle grows, they're stable, and definitely after they pass the critical size of nucleation, then they are stable even without the protein. So you can basically burn the protein afterwards.
I see. Have you tried doing that? Have you tried removing the proteins?
Yeah, we're drying it. I didn't try specifically, but we're drying it and the nanoparticle remains stable. But also the physics is pretty clear. And so once you pass a certain size, which could be calculated, a nanoparticle will remain stable even if you put it without a protein.
Thank you. That's very cool.
Did you have a hand up, Christian? Or was that an accident?
I did not. I think that was an accident, Lydia. I thought the talk was excellent, but no, I didn't have any questions.
Oh, okay. Awesome. Does anyone else have any questions for Amijai? No? Is there any way that we could help support you in your work?
Okay, so like I said, I personally moved to Sweden. There will be more work in the Baker Lab on minerals, protein. It's oriented more towards cement and let's call it non-electronic material. First of all, if anyone is interested more about it, I think the way to help is to open more channels of discussions, because as much as I talk to more people, understand how this could be applied and extended, and a lot of applications are not that easy to identify. Obviously, if I can make a GPU, a lot of people will be happy, perhaps with exception of NVIDIA, but the level of pristiness to make something like this is a bit crazy at this stage, at least, maybe some point in the future. So even understanding what kind of application could be done with this kind of materials will be great. So any discussion, any ideas, I would love to talk. Yeah.
On that note, I didn't put this in the chat because I was so busy on slide duty, but we do have a private Molecular Machines WhatsApp chat that I would love to add any of you to if you'd like to. So you all have my email. So shoot me one if you'd like to be added.
Yeah, sure.
Awesome.
Yes.
Yeah. Anyway, is there anything else from anyone? Tad.
Hi, this is a fun talk. So my question is, as you go about thinking about different kinds of, say, recycling or how difficult is the design problem for this, what you're trying to do? You're trying to replace certain different semiconductors. You mentioned GPUs, for example. So what kind of effort would it take to use it to design how you would use this technique as separate from the question of how you actually fabricate it? What's the challenge of the design challenge? You just get the.
Okay, I think I understood your question. So I think one thing is to design architecture. Some of it we know to do already, at least in the Baker Lab, but I think now but we're not totally able to do what nature is doing. So if you think about the bacteriophage, how complex it is, how many different proteins come together, this is something that we don't know yet. So there is
It had a few billion years to work on it.
Yes, maybe. But so there is work at the architecture level. A lot of things that I showed, like putting two chambers together, it's almost go. We are already doing this for the sake of it. I think the bigger challenge will be the templating. I think if I need to point out weakness in my research is that we found things that had reduced the energy needed to form the crystal, but I think we're at the local minimum. So I don't think we found really something that will be amazingly good template. And just as an example, so zinc oxide, in the paper, we used 3 millimolar. But zinc oxide can nucleate at 6 micromolar. So potentially we can find proteins that can template at 6 micromolar. If we find a protein like this, then you can really start controlling the reaction because the level of specificity become insane. And then you can even nucleate inside the blood, inside cells, because the concentration of zinc will be high enough. But I think the bigger, more challenging part is to focus on the templates, because shape we're doing for quite a lot of time in the Baker Lab and now in many other places. So I think the challenge will be to template different materials to know to work in a higher temperature, where the crystalline is higher in quality and so on. I hope I answered your question.
Yeah, thank you. It's good to have, I like to have a sense of how difficult is a design problem and how difficult is the manufacturing problem, and then to treat those as separate things. So yes, thank you.
Thank you so much, Amijai. Thank you all for coming, and we'll see you next month. Bye, everyone.